Okay, these are our objectives for our topic pala is replication. So this is the start of gene expression, no? So you have the topics on gene expression, replication, transcription, and then translation. So replication, these are the objectives. Oh, wala yung number one. Okay, anyway, number one objective is to be able to describe and give the different steps of replication. So you have the initiation, elongation, and termination. So number two is to describe and give the functions of the enzymes involved in the different steps of eukaryote replication. So we are emphasizing more on eukaryote, no? So alam niyo na siguro where it's almost the same sa prokaryotes, but with differences, no? And of course, the three is to explain the regulation of the initiation process, describe the elongation of the leading strands, and describe the process of termination, discuss DNA replication abnormalities and the different repair mechanisms. Okay? So this is our life, no? This is how our life goes. So this is your DNA. And the DNA is capable of synthesizing itself into two molecules of DNA. You know, so from one DNA molecule, two DNA molecules will be formed from replication. Okay, and one strand of the DNA. You know that the DNA has two strands, right? So one strand of this DNA molecule is used for transcription. So for the synthesis of RNA, so you have how many types of RNA? You have three types, no? Messenger, transfer, and then the ribosomal RNA. And from there, of course, the genes will be formed. Some will be formed in this process, and it will be translated into amino acids and proteins. That is how our life goes. Okay, so I know you are very familiar with that in your biologies. So replication is the synthesis of DNA for the provision of progeny with the genetic information possessed by the parents. Replication of DNA must be complete and carried out with maintain genetic stability with the organisms and species. And from one DNA molecule, two DNA molecules are formed. And each of the two strands of the DNA molecule are replicated simultaneously, but by different enzymes. Okay. So this is how it goes. Just raise your hands if you have any questions. So this is your DNA molecule, the parent DNA molecule, okay? We have two strands there. And they run anti-parallel to each other. So one strand runs at 5'3", the other strand runs at 3'5". So it should be opposite, no? And so one strand of these two DNA molecules, the two chains of DNA, are separated during replication, and each of the strand is capable of synthesizing another DNA, another chain of DNA molecule. So another DNA molecule finally, no? So you have from one strand, another strand is synthesized, so one DNA is formed, and from the other strand, another strand is synthesized, so the daughter strand, and so you have here another DNA molecule, okay? So how would you describe this kind of replication? How would you describe that?
Semi-conservative.
Yes, semi-conservative because one strand of the daughter strand is made up of the parent strand and of course the newly synthesized strand. Okay, so semi-conservative strand. because one strand with daughter DNA comes from the parent strand and the other is the newly synthesized strand, okay? So this is your parent DNA molecule, okay? How is the start of replication? Of course, it should start with the separation of the two chains, right? And as I mentioned earlier, one strand runs at 5'3", while the other strand runs at 3'5". Okay, so they're always anti-parallel to each other. And what makes up the periphery of these strands, of these chains? So this one is made up of your phosphates and sugar. Sugar, phosphates. What is the sugar in your DNA? What is the sugar in your DNA molecule? Anyone?
Deoxyribose.
Yes, ribose. Ribose sugar, okay? And you have two groups here. You have the minor group and major groups. What do you think occupies this major and the minor groups? The different proteins and your, ano ba yung protein niya? Excuse me. Stones, right? Histones and the other proteins. While your the core is made up of the what? The nucleic, the nucleotides. Nucleotides, and there are four of them. You have A, T, C, and G, and they are what? Complete. They should be complementary to each other. They should be complementary to each other so that your G is paired with a C, and your C again with the D, with A, and A with D. And if you notice, there are two hydrogen bonds between A and T and T and A, while in your CNG or GNC, we have three hydrogen bonds. Okay, you have to remember that. Okay, so this one is sugar phosphate moiety, and they run anti-parallel to each other. So, to change, okay, I already mentioned that. There should be complementary base pairing between A and T and C and D with utmost fidelity. With utmost fidelity, no? Ganyan kayo dapat sa mga girlfriend nyo, sa mga boyfriend nyo, with utmost fidelity, no? Gayahin nyo sila. So, again, this is the start of the separation, no? The separation of your the parent chains, the parent DNA chains, okay? So you have one strand, and this is the other strand, while this is what we call the what replicating fork. This is the replicating fork where the enzyme is usually is located, where the enzyme is located, okay? And it goes that way. This one is not yet open. It's not open yet. It should be the helicase that opens it, but in eukaryotes, there is another name for that. That not just helicase, but in eukaryote, it's called helicase, no? That separates the strands, the parent strands from each other. Okay, ayan. Okay, basahin nyo na lang yan. So here you are. Pareho ba yan? O, ano ba ito? Mabalik yata ako. So these are the features of replication. Be conservative, which we already discussed, no? We have described why it is semi-conservative. Why? Bakit bidirectionality? Bidirectionality. Okay. Okay, punta tayo sa sunod na in the next slide. And the origins are fixed. They have fixed origins, simultaneous strand unwinding and elongation, simultaneous strand. So, sabay, no? But one is continuous and the other is discontinuous. So we describe it as semi-discontinuous replication, okay? because one strand is replicated continuously, while the other strand is replicated discontinuously with the formation of Okazaki fragments. Okay, here. So this is a one-celled organism, no? We have here uh, separated na siya, no? This portion is already separated. And you have two replicating forks. Ito, because replication goes in both directions. So, opposite direction, no? So, kung ito yung ano, if this is the circular, semi-circular DNA, so one replication goes this way and the other one goes that way. Okay, kung north ito, the south yun, no? But there are two replicating forks here. There are two replicating forks. Okay, so this one, as they continue on with replication, then it is divided. That one-celled organism is divided into two, and at the end of which you have two DNA molecules. Okay, so this is the origin of replication, and that is there is a consensus sequence there that is usually the point of the origin. Okay, so these are the replicating forks or replication forks going that way and that way. This is going south, this is going north, and at the end of which you have two DNA molecules. But this is in prokaryote, no? One-celled organism. But it is different in eukaryote because eukaryotes have a it's not circular, but what? It's elongated, no? This one. So that means that the replication in eukaryotes, and because there are few proteins surrounding it, is fast. So replication is fast in prokaryotes, while it is slower in eukaryotes because of the presence of the histones and the other proteins. Okay, so what is the compensation there? There will be several origins of replication. Okay, so there will be several origins of replication, and this one of these bubbles is called the replicon, and in the same manner, it is bidirectional. Okay, bidirectional. So if this is the replicon, it goes that way, and this one is it goes this way, and this is the replicating fork of the first bubble, the replicating fork here and here. Okay, so becomes it compensates for its slow replication by having several organs of replication forming the replicons. Okay. Basahin nyo to. To make up for this difference, okay. Human cell replication begins at about 1,000 to 100,000 different origins of replication. Sa isang ano yan, sa isang DNA strand. Okay? So, bibilis-bilisan. So, another one, another example. And this is the beginning or the initiation of replication in eukaryotes. You have here the ARS. What is ARS? ARS is the autonomous recognition sequence. These are sequence of nucleotides that are recognized by the ORC. What is the ORC? Ano ba yung ORC here? Ito yung ORC. No? So the nucleotides here are the ARS. These are the autonomous replicating or recognition sequence, and it's usually made up of several 8 80s, 80s, 80s. Why do you think that is? Why do you think it selects, or rather the origin starts with a lot of aces? Anyone? 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 80, you know, 80 nucleotides, or yeah, 80 nucleotides. Oh, remember there are two strands here. So one is 80, 80, TA, TA. So why? Anyone? Because there are only two bonds, no? In 80s. Excuse me. While in DC, there are three bonds. So in between them, the hydrogen bonds, I mean, there are two hydrogen bonds between the 80s, A and P, and three hydrogen bonds between the D and C, so that it would be harder, no, it would be more difficult to separate uh, the GC because they are more bonded with each other, while the 80s only have two bonds. That clear? So again, this is your ARS. Ano nga ba yung ARS? Autonomous replicating sequence, right? And this is recognized by the ORC. This ORC, uh, origin of recognition, origin of recognition. Okay, so your origin of replication complex binds with the ARS. Okay, but that uh, is not strong enough to be able to separate the strands, or to function as a helicase. Okay, and this will be joined by the MC3, saan ba yun? Masyadong kita. MC, MC2 and MC7. MC7. Here. MCM2 and MCM7. Okay? But your MCM2 and MCM7, again, is weaker. It's weak. It cannot separate not function as an adequate helicase, but it has to be joined by CDT and CDC6. CDC6 from the cell cycle. Okay. Once it is bound by CDC6, then your MCM, once it has joined this, so your MCM will join your ORC, and your MCM is loaded with CDT6 and CDC6 and CDT, and it will become more potent and separate the strands from each other. That would be the initiation. That would be the initiation. So your MCM and your CDTs and CDCs are called the pre-replication complex. Pre-replication complex. Okay. If you have any questions, just raise your hands here. Okay. So yung ARS is composed of 11 base pair sequence. So there will be two MCM. There will be two MCM2 and MCM7 going separate, going to different directions. One is going south and the other is going north, so that there will be two replication forks that will be formed. Okay. So remember, or for your information, the initiation of the same site same site in the ARS, no, cannot occur until the end of mitosis. So what will prevent the initiation? Number one is the release of the ORC complex after its use and continued CDK activity until the start of mitosis, or phosphorylation of CDC6 and subsequent release, or degradation or inhibition by CDT1 by geminin. Inhibition of CDT1 by geminin. Okay, so please remember those uh, those things that can prevent the reinitiation of the already initiated ARS. Okay. So this is your antisense strand. This is the parent strand. Okay. And this is the beginning of the newly synthesized strand. Okay. Yeah. So how do these triphosphates enter into the chain so that the formation of DNA will become complete. Okay. So what enters into the chain would be a trinucleotide. So like cytosine trinucleotide triphosphate. Okay, a triphosphate pala. This is the incoming, and this is the new strand that is being formed, and this is the incoming nucleotide. Okay, please read this. Once the parent strand is unwound, complementary triphosphate nucleotides will enter into the elongating chain, and its 5-phosphate will bind with the 3-OH of the previous nucleotide in the chain by phosphodiester bonding, while its base will hydrogen bond with the base that is complementary to the nucleotide. Ito. Here. This is your parent strand. This is your incoming nucleotide. Okay? Ito yung primer. Ito yung this is the nucleotide that has been incorporated already on the chain. This is the incoming trinucleotide. Okay? So what will bind with, what is the bonding that occurs between the two, between the incoming nucleotide and the nucleotide previously? Okay, it's the phosphate part of the incoming nucleotide binds with the nucleophile, which is your OH of the previous nucleotide. You understand? Ito yung dati na, no? The first nucleotides that are incorporated into the chain, the daughter nucleotide, no, the daughter chain incorporated already, and this is the incoming nucleotide, and your incoming nucleotide binds with the OH through its phosphate. Yung unang phosphate, because there are 3 phosphates here, alpha, beta, and gamma. So the first phosphate will bind with the OH of the previous nucleotide, while ito na-cleave, no? This is released. The 2 phosphates are released and becomes a pyrophosphate, okay? There are 2 magnesium ions that take place in this particular reaction. So 1 phosphate will make this OH, or will deprotonate this OH so that it becomes more nucleophile. It becomes a more potent nucleophile, attract the pH, the P of the incoming nucleotide more, no? And the other nucleotide is helps in the cleavage of the two other phosphates. So helps in the release of the two phosphates. Okay, so ang natitira, what enters into the chain would be one phosphate, is the alpha phosphate. And what happens to the bases? What happens here? Ito, anong bonding ito? Anong bonding sila? Phosphodiester bond, no? Phosphodiester bonding. Ito naman, what is the bonding between the base of the parent strand and the incoming base? What is the bond here? Anong bond? Anyone? Anyone?
Hydrogen.
Come again? Come again?
Hydrogen.
Yes, good. Okay, hydrogen bonding. Okay. So yan ang formula yan, no? Once they hydrogen bond with each other, then you have a nucleotide that has been incorporated. But this one is ready again to receive the triphosphates, the nucleotide triphosphate that would be incoming. And that would be, the base here is base that enters, nucleotide that enters depends on the base of the parent strand. Now, if this is a T, then this should be an A. Okay? If this is a C, this should be a B. Okay? Wala na to, no? Na-explain ko na siya. Just read when you review. Ito same yan. Pareho din siya. Pareho din siya. And this is the product. The product, no? Yan. Yes? Any questions? Questions? Okay, these are the major proteins involved in replication, but this is for both single celled and for eukaryotes. Yes. So now that the strands are separated, parent strands are separated, and the elongation is starting. Let's go to the initiation. So this is the leading strand. Ito na, the starting, start of the synthesis of the outer strands, no? This is your strand that runs at T5, the parent strand that runs at T5. Now remember that the direction of the synthesis of the daughter strand is always at 5-3 direction. It's always at 5-3 direction, no? So what is synthesized continuously would be the strand. What strand will be synthesized continuously by the parent strand that runs at T5? Ay, sinabi ko na. Okay, this is the daughter strand that enters into the chain. This chain is the T5 chain of the parent strand. No? So it can be replicated continuously because the direction of your replication is at 5-3. Because we said that replication replication is at 5-3 direction. Only at 5-3 direction. No? Hindi pwedeng T5. So hindi mo pwedeng ma-replicate yung 5-3 that continuously, no? Kasi they should always be what? Anti-parallel to each other. Remember? That two chains should be anti-parallel to each other. So remember that this is the parent strand that runs at 3,5 is continuously replicated, and this is called the leading strand. While the other strand is the strand that runs at 5,3. Ito, parent strand that runs at 5,3 cannot be replicated at 5,3 direction. No? So it has to go into the opposite direction in order for it to become synthesized at 5,3 direction with the formation of Okazaki fragments. And this is called the lagging strand. This is called the leading strand. Okay? Again, read that while. So this is the direction continuously because the parent strand it runs at 3,5 while the parent strand that runs at 5,3 is replicated with the formation of Okazaki fragments. Okay? Now the enzyme should always be by the replicating fork. The enzyme is always at the site of the replicating fork. Now, this is your cell cycle, and DNA replication occurs during the S phase of the cell cycle, which takes place at for 6 to 8 hours. And the different cyclins that play important roles in the initiation come from the cycle, come from the cell cycle, like the CDT, the CDC, DC6. Okay. Again, these are enzymes for both prokaryotes and eukaryotes. So this one is enzymes that are functions for eukaryotic DNA polymerase. No? So Pol A replicates in the initiation in conjunction with primase. Okay, and it has a function for DNA repair. Okay, while polymerase B beta is solely for DNA repair. Polymerase gamma for DNA replication in mitochondria alone. Mitochondria lang siya. While polymerase delta, replication of DNA synthesis, the leading and or the lagging strand. Now, sometimes different books say different things, different enzymes. Si Devlin says that for for the uh, for the leading strand, it is replicated by the delta, and the lagging strand replicated by epsilon. So both of these have polymerase delta and your epsilon. They have DNA repair mechanisms. Okay. So again, you have here the two strands that have been separated. This is the 3,5 strand, so it is continuously replicated at 5,3 direction. Okay, this is the position of the enzyme. While the lagging strand, which is the parent strand that runs at 5,3, is replicated with the formation of Okazaki fragments. An Okazaki fragment is made up of RNA and DNA. So that's is what we call your Okazaki fragment. Okay? So ito, How is it replicated 5-3 direction? In this particular area where the enzyme is, the parent strand runs coils around each other, forming a parang trombone, no? Ito. This is the parent strand, that 5-3 direction. It coils around itself. Tapos it is replicated here. So baliktad na, no? It can now be replicated at 5-3. Not replicate, it can be replicated at 5-3 na. And it is thrown towards the end. Yung una, the first, this is made up of your RNA, DNA, adducts. No? RNA, DNA. Lahat yan, RNA, DNA, RNA, DNA. It is pushed towards the end. Then the next one will be pushed here. Susunod siya. And so on. Until the whole strand is replicated. So in the lagging strand, again, the parent strand, the strand that runs at type 3, forms a coil or a trombone form in order for the RNA and DNA will be replicated discontinuously at 5-3 direction. Again, it is thrown towards the end. Ganyan-ganyan siya, no? So ito, pagdating dito, wala na siyang enzyme, no? Dito lang yung enzyme. Okay. Any questions? These are the parent strands that have not been opened, no? Pareho lang yan. Now, what is it topoisomerase? Topoisomerase is an enzyme that it's an enzyme located here. Here. Why do you think there should be an enzyme that should be present here? Because if this is open, from here to there, and chances are there will be stress here. Huh? The end part of that coil of that end part of that of DNA molecule will have torsion or will become stressed, and there will be what becomes stress. There will be torsion. Huh? And so you have your topoisomerase to create nicks in order for the torsion or the stress be eased, no? Is the torsion or the stress that is created by the opening of the strands. That is your helicase. Oh, sorry. That is your isomerase. Isomerase, that's isomerase. Oh, I'm sorry. Topoisomerase. Here. Topoisomerase. Because that will create portion here. That will create if if the strands are separated from each other continuously, there will come a time when torsion is created at this part of the chain. So again, the start of replication. Yung start natin, no? It's going there. Pag ganon. Ito naman, pabaliktad, no? So, the first nucleotides that were incorporated in both strands are not DNA, but RNAs, RNA molecules. So why is that so? Because DNA needs a an available OH in order for it to be attached, no? Kailangan niya ng available OH. So, kaya RNA muna siya. RNA doesn't need that. Okay? So, after 10 RNA molecules, then DNA follows. So, the first enzyme that is responsible for the synthesis of the RNAs in the initiation is called the primase. Primase in both. So, each fragment, each DNA, each Okazaki fragment consists of meron din siyang primase, umpisa, and RNAs muna yan. So RNAs and DNAs. Edit RNAs muna, mga 10 to 10, siguro mga 10 to 20 RNAs. And then the other enzyme for initiation would be your polymerase A. Polymerase A, which will now synthesize the first DNA molecules. Then, so this one, in the same manner, the first nucleotides that are incorporated into the Okazaki fragments are RNA, and then through the same enzyme, the primase, and the polymerase A, and then followed by the polymerase D. Polymerase E pala. Polymerase E for the lagging strand, and polymerase D for the leading strand. So yun ang gagamitin natin, no? Polymerase D for the leading strand, polymerase E for the lagging strand. Okay? So palagi yan, dito, for every Okazaki fragment, you have polymerase, primase muna, primase, then polymerase A, followed by the polymerase delta. Okay? For the rest of the nucleotide, for the rest of the DNAs are synthesized. But once it reaches the end, once it reaches the nucleotide that is already there, that is present, then it becomes the DNA, or rather the DNA, the polymerase epsilon gets out of the strand, and again you have your primase and you have your polymerase A, and then followed by ramitor, followed by polymerase D. Pag nag-polymerase D na, then enzymes polymerase A and the primase will be removed. So here, now after the strand has been separated, ito, that would be, the start would be here, no? But dito ngayon, so hanggang dito na yung replication. So dito na, this is the separated strand. In order for them not to re-anneal, or ibig sabihin, not to come together again, because they are so attracted to each other that pabayaan mo sila, they will be attracted again. They will be attracted to each other, no? So in order for the annealing to be inhibited, you have this RPA. Ano ba yung RPA? Replication protein A, no? Replication protein A. So that will prevent the re-annealing of the single-strand brand. In prokaryotes, you call that the single-stranded binding protein. So in eukaryotes, you call that the RPA, or the replicating protein A. Okay? Any questions here? Ito yung topoisomerase mo that will prevent torsion. Torsion ang tawag doon, stress or or ano pa yung coiling, no? Coiling. And it creates a nick. Nick. Alam mo nung nick, ano? Parang kinat siya ng konti para to prevent stress. Ease the stress that is going on. Now, this is your polymerase. Polymerase delta. Polymerase delta for the leading strand. Now, what are these? This is PCNA. Ano ba yung PCNA? Ano ba yung PCNA? Anyone?
Proliferating cell nuclear antigen po.
Come again? What is that?
PCNA stands for proliferating cell nuclear antigen po.
Okay, polymerase cell nuclear antigen. So what is the use? Why is it bound to your polymerase delta? In order for the enzyme to have processivity. What is processivity? Processivity means continuous synthesis of molecules, of the DNA molecules, no? From here to there, continuous siya. Hindi siya, because if there is no processivity, then one nucleotide, after one nucleotide that is synthesized, then it will be released from the chain, no? In order for it not to be released from the chain, so that it can continuously synthesize, you have this PCNA, okay? That cleaves, or rather that is bound to your polymerase delta. And even in polymerase epsilon, meron din siya. Now this one is the what do you call this? RFC. What is RFC?
Replication factor C po, Doc.
Replication factor C. So do not confuse that with RPA, no? Ibang function nito. This is the clamp loader. Now this is called the clamp. Huh? This is called the clamp. Yung PCNA is called the clamp. And this is the clamp loader. It loads the clamp into the strand, into the chain. It loads the clamp into the chain. Yung RFC. Okay, do not forget the names of the enzymes. Kasi importante yan. Okay, ano po bang enzyme hindi natin nakuha dito? Oh, RNAs. Now, RNAs and FEN1 are responsible for the release of the RNAs. Remember, your RNA should not really be a part of your DNA molecule, right? Should be DNA and the RNA. So it is released by your RNAs. Most of the RNAs are released by your RNAs, while the last RNA, kasi ito may tinitirang isa yan, ha? May tinitirang isa. It's released by your FEN1. FEN1. What is FEN1?
Flap endonuclease 1 po.
Okay. FEN1. So again, the RNA molecules that are present in your here in the Kawasaki fragments are released by most of them, no, are released by the RNAs, and the last one, because ito tinitirahan niya ng isa, no? Last one is released by your FEN1. Okay? What is the function of your DNA ligase? DNA ligase. Ligates one Okazaki fragment to the next Okazaki fragment. Remember, they are not continuous, right? So once it reaches the one Okazaki fragment reaches the last one, o dito. Once this one reaches this one, then it is ligated. Pero walang putol, no? Ligated by your DNA ligase. Any questions dito? Any questions? Yan. So 10 RNA muna sa primase, no? 10 RNA, and then your polymerase A takes over and synthesizes 20 to 30 deoxyribonucleotides. And then after that, this will be continued by your polymerase, either polymerase delta or polymerase epsilon. Okay, another example. Magulo to, no? Mahirap intindihin. Well, anyway, this is your, ito yung nagko-coil siya. So your Okazaki fragment is made up of RNA and DNA. This is your pump. This is your enzyme. Nasaan na yung loader? Wala na, no? If you're loading, then that pump loader gets out. Okay? And here is your coil. Ito. Naging siya. So your synthesis should be here at this point. So, ganon. Galing pala dito. Please read this, no? For your review. These are the cyclins that are found in your cell cycle, cell cycle. Another one. Any questions? Any questions? So remember that your polymerase delta and epsilon have the 3,5 nuclease proofreading activity. So it can repair, no? It's capable of DNA repair if ever there is mutation in order to ensure accuracy. So that is why you see very little, wala na yan, same, yan, same, this is, very sure, DNA ligase. So after your one Okazaki fragment, this is the next Okazaki fragment, then these are ligated so that there will be no cuts. And again, what is the bonding? What is the process by 5 phosphodiester bonding? Yes. Oh, we'll talk about polymerase. Now these are the repair mechanisms if there are abnormalities. Mismatch repair, mismatch. So you can understand that if different nucleotide is incorporated into the chain. So copying errors, so single base or 2 to 5 base and repaired and paired loops. What is the solution? Strand cutting or exonuclease digestion and replacement. So, ano ba yung enzymes doon? Yung polymerases, no? Ano ba pag-alis nito? Okay. Base excision repair. So, yung base ang nawala, no? Spontaneous chemical or radiation damage to a single base. So, base removal. Base removal ang solution. Okay. Removal and replacement of the base. Any questions? I can hear somebody talking. So if you want, yes. Any questions? Do you have something to say? I can hear somebody talking. I cannot understand. Is that you? I mean, the class? Is that one of you? If you're not asking questions, if you do not have comments, please mute yourself. And this is nucleotide excision repair. The whole nucleotide is damaged. This is by spontaneous chemical radiation damage by DNA segment, no? And being destructed. Sino ba yun? If you want to ask, please ask na. Mute. Base removal by, so the whole base is removed. Oh, ay, ito pala na tayo. Nucleotide excision, removal of approximately 30 nucleotide oligomer and replacement can be. So what enzyme will function for this? You have the different polymerases, no? Yung polymerase delta and polymerase epsilon, and even the primase. Even the primase and the polymerase A. And this is double strand repair by ionizing radiation, chemotherapy, oxidative free radicals. So, unwinding, alignment, ligation. These are the repair mechanisms. Now, how is cigarette smoking unhealthy? So this is your benzoapyrene that is present in cigarette smoke. It's not carcinogenic by itself, but once it is oxidized, it can react with guanine, the DNA molecule. So it can react with guanine and bond with your guanine, the NH group of your guanine, and of course this will interrupt, or this will inhibit the progress of replication. And this is by what? What is this? By ultraviolet rays. So if there are two thymine DNAs that are thymine nucleotides that are side by side each other, no? If the person is exposed to ultraviolet rays, then this thymine dimer or the cyclobutane ring can be formed. Ito, cyclobutane ring. So nagkaroon siya ng bonds between the two thymine nucleotides, and it will also inhibit the progress of your replication. Okay. Now, what is the telomere? After the removal of the RNA primer here, in the three portion of the DNA, or rather of the strand, no, the three portion. So this is removed by the polymerases. What will happen is this one will overhang. Wala na siyang partner, no? There will be no partner. And this happens for every replication. In every replication, this happens. You call this the telomeres. Telomeres. So that would be TTGGAA. TTAGGG. TTAGGG. So if that continuously occur without any repair, then what will happen is for every cell division, your DNA molecule will become shorter every time cell division occurs, no? But that is ay, ano ba yun? Where is that? Here. So this is your TTAGGG now overhanging after the RNA is removed, no? After the RNA is removed, at the three end of the strand. So you have the telomerase, that's the enzyme that consists of RNAs, several RNA molecules. And so this will attach itself, and this will start to synthesize the appropriate DNA in this particular strand. This only occurs, your telomerase only happens in cells that are fast dividing, no? Cells that are dividing fast. What about your somatic cells? What is the division there, cell division? Mabagal lang, no? Mabagal lang cell division yan. So, but in, bigyan mo nga ako ng example of a fast dividing cell. Cell that is speedily dividing. So that contains the enzyme telomerase, no? Ano ba example nan? The embryo, no? The embryo is a fast dividing cell, no? And it contains telomerase. So if there is any gap there, it can also always be repaired by the enzyme telomerase, because this contains the telomerase and reverse transcriptase. Reverse transcriptase, wherein the RNA is capable of synthesizing DNA. So these are RNAs in your telomerase. These are capable of synthesizing DNAs that can become bonded with DNAs in the other strand. Okay. Now another example is of a fast dividing cell is what? Cancer cells. Cancer cells. Cancer cells are actually what? Are actually they do not really die, no? because of the presence of your telomerase there. So, tuloy-tuloy ang division niya. That's why it grows so fast. Cancer cells grow so fast. Ano pang iba na fast ang division? Now, what is the use of this in our time? In stem cell, no? Stem cell. This is present. Telomerase is present. So this is why you have your stem cell therapy. Rich people go to certain hospital for stem cell therapy because of this. To make them younger or reverse a particular disorder, disease, no? So, yung mga mayayaman lang ang kumul niyan kasi masyadong mahal daw yung stem cell therapy. So, don't believe those kahit saan na lang na merong nagbibigay ng stem cell. Okay, these are not true, no? So I think that would be the entire. Please read this. Okay. So any questions? Any questions?
No po daw.
No questions? Good. Intindihan nyo? We will have our quiz on kailan bang quiz natin dito. So then we will go to the wala ako. Here. Stop share na lang ako, ha? Okay. So I will share again this. Hmm. Can you see? There.
Yes po,
Yes.
Doc.
You can see the questions? Kita nyo? Ano ba yun? Bakit nawawala siya?
Meron po.
Meron ba? Ako hindi ko nakikita. Okay. So who can answer number 1? So just describe and its interaction with ORC. Point out the ARS in a replicating eukaryotic cell and describe its interaction with ORC. Anyone? 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 Sabihin nyo na lang, what is ARS? And how is it related to the ORC? Anyone? Akala ko wala kayong questions.
Doc,
Yes.
to answer very concisely lang po,
Yes.
ARS stands for autonomously replicating sequences, and it functions as a binding site for ORC, which is the origin recognition complex. So what ORC does is that it binds with ARS to direct DNA replication po.
Okay. So what is the composition of ARS usually?
An 11 base pair sequence po.
Of what nucleotides? Apat lang naman yung nucleotides, di ba? So, what nucleotides? Anyone?
Dok, ano po, adenine and thymine po.
Adenine and yes.
Thymine.
Okay, so 80, no? 80, The reason for which is cause what? Why not GC?
Kasi po yung adenine and thymine, dalawa lang po yung hydrogen bonds niya. So it's easier to break down po.
Okay, good. So number 2, please. Number 2, we can answer number 2. Describe the function or tell me the function of MCM2, MCM7, and replication fork. The function of MCM2, MCM7. Yeah. And siya. So what is the function? What is the function? It functions as the helicase or what? Function as the helicase? Oh, in prokaryotes it's called the helicase, but in eukaryotes it's called, this is the name, or these are other the proteins that separate, oh, that separate the strands. But as I said, as we said, it's not potent yet. It's not yet capable of really dividing the strands because it is not strong. It's weak. Its protein is weak. So then it has to bind. It has to be bound to CDC6 and CDT1. Okay. Once this is called what? These two are called pre-initiation complex. Pre-initiation complex. Okay. So once that is your MCM is bound to these two, then it will start to unwind the DNA molecule. And each MCM complex goes into different directions. Okay? Next question. Next question. What is the function of pol A and primase, the elongation in both the leading and the lagging strands? Anyone?
RNA primer synthesis po, Doc.
Okay. So they synthesize the primer. So composing of what?
10 ribonucleotides po.
Yeah, what nucleotides? RNA, DNA?
So for the primase po, it will have ribonucleotides po.
Yes, the primase. And what about the pol A?
For pol A po, it will replace the yung ribonucleotides from the primase with deoxyribonucleotides po.
Oh, hindi pa niya it will not be released yet, no? Only after the synthesis of the DNA, okay? And by the polymerase A. So, number 4, describe how the lagging strand is elongated in relation to the replicating fork. Anyone?
Doc,
Yes.
the lagging strand po, unlike the leading strand, is elongated in the direction opposite the replication fork. Kaya the the one that what is produced is Okazaki fragments kasi it's discontinuously produced. So like the similar din naman po yung process sa leading strand, only that because it's the result is Okazaki fragments. It needs DNA ligase to seal the nicks in between the different fragments. And if I remember po correctly, each
Yes.
Okazaki fragment needs a new primase.
Yes, each Okazaki fragment. Because it's discontinuous. It's discontinuous, no? It's one Okazaki fragment is not really connected with the other Okazaki fragment. But once it reaches that part, the last nucleotide that's been incorporated already, then it will hydrogen bond and remove the RNA. Okay, so it needs a ligase after the removal of the RNA the and the incorporation of the new DNA molecules. Okay. Next question. Ano na ba tayo? How are the leading and lagging strands stabilized so they can continue to function with processivity? So, paano siya na-stabilize? Because it will not be released after one nucleotide is synthesized. So that would be continuous replication of the daughter strand that is called processivity. So how are they stabilized? What is that protein that is incorporated into the polymerase in order for it to become or to have processivity? So either polymerase delta or polymerase E needs this in order for processivity to function. I mean,
Doc,
in
is
order,
this po, Doc, for this
yes,
po, would it be the sliding clamp? So that's the proliferating cell nuclear antigen.
the sliding clamp or what is the name of
NA po, proliferating cell nuclear antigen.
Yes, that is correct. Yes, so that is the protein that stabilizes that the enzyme so that processivity will proceed, no? Okay. Describe the termination of replication in eukaryotes. Paano ba na, how is it terminated? Sabihin mo yung enzymes. What are the enzymes?
Telomerase po.
Come again?
Telomerase.
No.
Ligase po.
DNA ligase, Doc.
No. Termination of eukaryotes. Termination of replication.
Topoisomerase po.
No, hindi. Ang topoisomerase is located at the parent strand that is not yet separated, no? So well, I was thinking of the FEN1 and the RNase, no? RNase. Pero hindi naman, siguro kasi hindi nyo maintindihan. Hindi siguro clear yung question, ano? Termination. Okay? Sige. Anyway, yun na rin ang termination niya, no? Explain how nucleotide excision repair is brought about. Nucleotide excision. So, buong nucleotide. Nucleotide. Yan. One.
For this po, magkakaroon ng removal of approximately 30 nucleotide oligomers and then it will be replaced po with the proper
Yes.
nucleotides.
Okay, just read on this, no? Andiyan naman siya. Nakatabulate siya. Then, describe the DNA damage, but cigarette smoke. So, how is the damage that's brought about by cigarette smoke?
Yung, Doc, yung benzopyrene po pag na-oxidize, magiging carcinogenic.
Yes. May binding with what? Guanine, no? Guanine sa,
One yung residues po.
yes, DNA molecule. And how does ultraviolet rays damage DNA molecule? Excuse me. Ito. Exposure to ultraviolet rays. Formation of what? Diamond dimer.
Doc.
Dimers, Doc.
Ay,
Or cyclobutane ring. Diamond
Cyclobutane.
dimer na lang. Okay. Ang repair niyan is lyase. There's an enzyme that repairs it. It's called the lyase. 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 Okay? Hindi ko nasulat. Okay, just remember that. And the 12 number, tabulate the different enzymes involved in initiation, elongation, termination. Who will give me one enzyme or kahit na dalawa for the initiation?
Doc, helicase po.
Helicase, please, some eukaryotes. Ano pangalan niya sa eukaryote? MCM. MCM. Huh? MCM siya. What's the function?
Responsible for unwinding po the two DNA strands to separate them.
Yes. Who else? Another enzyme?
Primase po.
Primase? Yes. Function?
It initiates synthesis of RNA primers po.
Okay. Another one? Another enzyme for initiation?
Gyrase po. Topoisomerase.
Topoisomerase? That's sa elongation na yon. That's for elongation. Okay? Prevents the supercoiling or what you call the torsion of the unwound chain, unwound parent chain. So, elongation na yon. So, ano yung enzymes dito? Ano pa? What is that enzyme or protein that prevents reannealing? Prevents reannealing.
Replication fork.
No?
RPA.
RPA. RPA? RPA? Yan natin. Pero, RPA nga, no? So, what's RPA?
Replication protein A.
Okay. And that would prevent the reannealing of the two chains, the two parent strands. Ano pa? Is there another enzyme? Is there another enzyme that we have missed? Wala na? Okay, elongation. Elongation.
DNA polymerase po.
Polymerase what? There are two strands that are replicated by different enzymes during elongation. Ano yun?
Delta and epsilon po.
Come again?
Delta and epsilon.
Okay. Function?
Add nucleotides po to the strand growing. 5' to 3' po.
Anong tawag mo doon sa enzyme?
Leading strand po.
Ano pa? Ano pang ibang enzyme?
Topoisomerase to.
Topoisomerase, okay. Function?
Relieves torsional strain po para matanggal po yung introduce po yung negative supercoils into the DNA molecule.
Okay. Other enzymes, other proteins?
Ligase po.
Ligase, okay. Ano pa? Aside from the enzymes, what is needed? Aside from the polymerases, what is needed?
Sliding clamps po.
Yes, sliding clamp or the PCNA, right? Ano pa?
Yes
May
po,
isa
Doc.
pa. What is needed for the sliding clamp to bind with a strand?
RFC po.
RFC. Okay. Termination. Termination, ito lang yung RNAs and what? RNAs and
10-1 po.
N1. N1. Okay. Any questions? Any questions? We'll only have 5 questions for the quiz. Shall I stop sharing? Okay. So do you have any questions? Do you have any questions? Replication?
None po, Doc.
Come again? None? Oh, very good. Okay.
Yes po, none po.
So I will expect you to get all the 5 points sa quiz. Okay, madali lang naman yung replication eh. Right? Kailangan alam mo lang yung mga enzymes, elongation, initiation, elongation, termination. Okay? So, Miss, andito na ba kayo? Miss Custodio? Or Miss Katibog? Custodio, are you here? Wala. Miss Cueva? Miss Cueva?
Hello po, Doc.
Hello, andyan na ba sila? Andyan na ba si Miss Tibog? Wala. Miss Katibog? Are you here? Miss Custodio? Wala sila. Vicky? Vicky? Vicky, are you still here? Wala na yata si Vicky. Or anyway, Miss Cueva, sabi na lang sa kanila, no? Please tell them that I will be available tomorrow at the office. I will be there from 9 to Siguro until after lunch. Okay?
Yes po, Doc. Noted po.
Okay, sige. Thank you. So if you do not have any questions, then please get ready for your next activity, which is what? Ano ba yun? Plenary saan? With Dr. Lee Reyes, no? Sige. Okay? So